next up we have Dr. Shah from the Division of Cardiology. She's going to speak to us today. We sort of put down a tentative title, and I don't know if your slide actually has the exact same title. No, novel biomarkers for predicting cardio. Yep, it's on. Yes, biomarkers. So everybody, please welcome. You can advance here if you want, and there's a laser pointer.
Okay, great, thank you. Thank you for having me. I'm sorry to mess up the schedule. I was stuck in a procedure. What John asked me to speak about today was an R21 application that we submitted, a collaborative R21 application that we submitted a couple of weeks ago in response to, there's actually 3 RFAs from the NHLBI to support research that sort of looks at the intersection between HIV and cardiovascular disease. This R21 application was a basic science R21 application, and so what I thought I'd do is present some of the preliminary data really on the cardiovascular side that led us to put this joint application in, tell you a little bit about our specific aims, we'd love some feedback, and then sort of talk about potential next directions. Sorry, I want to point out that this really was a collaborative effort. I had so much fun with this, working with John, Eric Velazquez from the Echo Lab, Lance Okeki, is Lance here? Fabulous fellow, really a very talented individual, Susanna Nagy and Mary McGeller with NID. So a really fun group to work with, and we got a lot done in a short period of time. I'm just going to skip over that. So my lab, people call me a translational researcher. I'm a cardiologist. But my work has really been interested in looking at biomarkers of cardiovascular disease. I was trained originally in genetics, we thought we were going to solve many of the cardiovascular ailments by looking at genetic markers, and what we realized right when I was starting my career is that the genetics of common complex diseases were, interesting, but they weren't really going to identify biomarkers that explained even any of the heritability of cardiovascular disease, and certainly not clinically useful biomarkers. And so I was lucky enough, to be at a place like Duke and to have lots of different resources available to me to sort of say, okay, well, maybe it's not just going to be DNA-based variation. Cardiovascular disease, regardless of what the sort of starting signal of cardiovascular disease, is a temporal process over the course of somebody's lifetime. And to imagine just looking at static DNA variation right when somebody's born and say this is your risk of cardiovascular disease is somewhat of a naive, I think, view to take. And especially when we're looking at human disease where there's a lot of noise from a lot of different places, that maybe you could look closer biologically to the signal by looking at circulating biomarkers that are not DNA-based, so biomarkers that change over time and may be more proximal to the disease process and relevant, hopefully, to the disease process. And so for those of you who kind of don't live in this omics world that we talk about, just to remind you that we can look at lots of DNA-based variation, and now people are doing whole genome sequencing where you can look at, all 3 billion nucleotides. The technology has really evolved so you can look at transcriptomics. This is the idea that not all DNA is constantly being transcribed into RNA transcripts, and so you can get snapshots of what's active in the context of the DNA footprint, blueprint, at any given point and do transcriptomic analysis. Transcripts then get translated into proteins, so you could look at proteomics, and we do some proteomics, but I won't really talk about that today. And what I really want to focus on today is metabolomics, the sort of enzymatic byproducts of all these different reactions. The way I think of metabolomics, it's sort of the biochemical byproducts of all the sort of cellular machinery that occurs throughout your body, and the idea that metabolites could serve as sort of reporters of all of these molecular processes kind of combined into one. I think Rob Califf describes metabolomics pretty well. He says it's like looking in somebody's garbage the morning after to see how the party went the night before. So like sort of this idea that, your body is constantly secreting these waste byproducts in the blood and that you can pick these up. And to keep in mind, these are sort of at micromolar, nanomolar levels that we measure in the blood. Or you can measure in any tissue. We happen to do it in blood because in cardiology, most people don't want to give us their hearts and their aortas. And just to put a shout out again to the fact that Duke is such an amazing place to be that we have a newly formed Duke Molecular Physiology Institute. Through a lot of support from Duke leadership and Mary Klotman, we were able to form this institute, which combines sort of basic science, translational research, and clinical science. So we have core facilities for metabolomics, gene expression, genomics, computational biology, and Duke renovated a bunch of tobacco warehouse space for us downtown. So we actually have a sort of a physical presence where we can do basic science, translational science, and clinical science in this omics realm all in one place. And this is led by Dr. Chris Newgard. So it's been a great resource for me as a physician scientist. So just, if I could just present a little bit of preliminary data just to kind of give you a flavor of where this R21 came about. This is really work purely in cardiovascular disease, and hopefully I will explain to you what the relevance potentially might be to cardiovascular disease in persons living with HIV. We have a large biorepository here at Duke where we collect blood on everybody who comes through the cardiac catheterization lab. So patients who are being evaluated for clinical concern of ischemic heart disease, they come in for a heart angiogram and we collect blood on them. We get over 600 clinical variables when they get enrolled, and then we follow them yearly after that. And so we have up to 13 years of follow-up on these people, almost 10,000 people in the biorepository. And this is just sort of a Venn diagram showing you we have lots of different molecular data through different grants that we've been able to generate in this biorepository. One thing that's been particularly exciting for us in the work that we've done is we basically said, well, can you look at circulating markers, these circulating metabolites, these sort of the trash that your cellular metabolism is kind of putting out? Can you measure these circulating metabolites, and do they report on cardiovascular disease at all? And what we found, we have a few papers in all different sort of cardiovascular phenotypes, but the one that was kind of really exciting for us in this sort of translational realm is we found a set of metabolites that actually predict who's going to have cardiovascular death or heart attack in the future. So this can be up to several years in the future. We collect their blood when they come in, and we found, this is sort of using unbiased approaches, meaning we didn't focus on one set of metabolites, we just said which metabolites predict events. And it was this set of short-chain dicarboxylacyl carnitines. And what we found was that the higher, this is the blue is the highest tertile of these metabolite levels, the higher your levels of these metabolites right now, the higher your risk of having a heart attack or dying of cardiovascular disease in the future. And I initially thought it was sort of statistical aberrancy because we found it in a very small data set, and then what we found is actually replicated in several different data sets. The good or bad thing, however you want to look at it, is very little was known about these metabolites, and in fact there still is not much known about these metabolites, but the good part is that, it gives us research to do, and hopefully I'll show you what we think might be going on with what these metabolites are reporting on. So really very little known about the source of these metabolites, the biological pathways they may be reporting on. Skip that. Sorry, I'm going to skip all of these. What I decided to do was to say, okay, well, let's do a genetic screen. So I've always been interested in biomarkers of cardiovascular disease, interested in genetics. Well, maybe if we use these metabolites themselves as phenotypes for our genetic studies, maybe it'll identify new genes for cardiovascular disease, and importantly, potentially help us understand what's the genetic architecture that regulates circulating levels of these metabolites, and potentially tell us what's the biology underlying these metabolites. And what we did here was just a small study to show that the metabolites are heritable, which in fact we found that several of these short-chain dicarboxylacyl carnitines are very heritable, in fact more heritable than traditional cardiovascular risk factors. And then we did an unbiased genetic screen, so we sort of said we have no idea, we're agnostic to what might be regulating levels of these metabolites. And we did a genome-wide association study in almost 4,000 people where we said, what are the genes that are regulating levels of these metabolites? And what we found were 6 different genes that were genome-wide significant for being associated with levels of these metabolites. And each individual gene is kind of interesting, but I think what's really interesting, to me anyway, is that all 6 of the genes report on endoplasmic reticulum stress, so reporting on ER stress. So we thought that was kind of provocative that, in this sort of, million SNP screen that the top 6 genes were ER stress genes, and perhaps maybe that was a little tidbit that these metabolites might be reporting on ER stress. And then taking a totally different approach, we looked at epigenetics, and we said, well, are there epigenetic markers that differentiate patients who have high levels of these metabolites versus those who have low levels? And not to belabor sort of all the, I think fun, but other people find boring, statistical details of how you analyze this data, we ended up with 7 candidate genes that really were differentially methylated, meaning showing epigenetic changes in patients who have high versus low levels. And in fact, 2 of these genes, and including the most significant one, were ER stress genes, and specifically pointing to the ubiquitin proteasome arm of ER stress, which I'll tell you about in a second. So 2 platforms that independently in an unbiased fashion seem to suggest that these metabolites may be related to ER stress. Then a third platform, we actually looked at peripheral blood gene expression. So we looked at microarrays, we looked at thousands of RNA transcripts, and in an unbiased fashion basically said what RNA transcripts are differentially expressed based on what your levels of these metabolites are. And this is all unbiased, sort of fancy statistical work that we do, where we don't tell the computer what we're thinking, and the computer kind of takes into account what it knows, what we know about biology from in silico databases and sort of these unbiased analyses. And in fact, there were 3 pathways that came out as the most statistically significant in terms of how differential expression was reporting on these metabolite levels. And the 3 pathways were the proteasome pathway, again, this ubiquitin proteasome arm of ER stress, cardiac muscle contraction, which we always like to see. And then I guess initially I was a little bit disappointed because Parkinson's disease came up, and that didn't make any sense. But when you actually look at the genes that live in the KEGG pathway for Parkinson's disease, almost 2/3 of them are actually ER stress genes, because ER stress is known to be a significant pathogenic mechanism of Parkinson's disease, and actually several diseases. So our sort of working model is that there is genetic and epigenetic regulation or epigenetic variation that predisposes to ER stress or dysregulated proteasome function, increased ER stress, that the ER stress is reflected by these higher levels of these metabolites. So we don't think the metabolites are actually causing the disease, but they're reporting on people who have heightened ER stress. The metabolites themselves predict cardiovascular events. And then what was kind of cool is we took the genetic variants that were associated with the metabolites and showed that they themselves also predict cardiovascular events. And I don't have the data, but we just finished some in vivo studies in kidney cells where we actually showed that when you induce ER stress with tunicamycin, that you in fact see a concomitant increase in these metabolites along with ER stress markers. So just to quickly remind you guys, I don't know how much knowledge there is about ER stress in the room, maybe more than I have, but that the endoplasmic reticulum is a multifunctional organelle that basically takes care of all kinds of protein stuff. One of the main functions of the ER is actually to take care of bad proteins, so misfolded proteins, excess proteins, any kind of proteins, but also if you have excess proteins in the context of hyperglycemia, dyslipidemia, so you can imagine with these sort of cardiometabolic risk factors, that's how you handle those proteins. So it takes these proteins, it sticks a ubiquitin tag on it, escorts the protein to the proteasome for degradation. If you have too much misfolded proteins or excess proteins, it induces ER stress, which initially can be a beneficial thing to happen because it leads to basically you handle the proteins. It can lead to autophagy, but then eventually the cell says, this is too much, I can't handle this, and it leads to apoptosis as opposed to autophagy. So it's really a balance for ER stress. I promise I'll take it to HIV. So this is sort of the work that I've been doing for the past couple of years. Again, in collaboration with a lot of the infectious disease doctors, there was a growing interest in really understanding mechanisms of cardiovascular disease in HIV. And it turns out there's actually potentially a role for ER stress in HIV, and there's been some preclinical studies. Protease inhibitors specifically actually trigger oxygen species generation, which leads to apoptosis, mitochondrial dysfunction, and this ubiquitin proteasome system dysregulation. It's been shown that the HIV virus can cause gut inflammation, and this is potentially due to induction of ER stress pathways. And then this was kind of a small study, but that ER stress markers have been shown to be upregulated in patients who, in brain tissue from HIV-infected postmortem patients. And so there's some provocative stuff to suggest that there's a role of ER stress in primary HIV infection and potentially with HIV treatment. And sort of led us to the hypothesis of, can we sort of use this model to really sort of understand, I think, biomarkers of predicting cardiovascular disease and cardiometabolic abnormalities in patient, I learned the proper terminology, persons living with HIV. And hopefully what I've been able to show you is that we identify biomarkers, but it also helps us understand mechanism often also. So the R21 we ended up putting in was really capitalizing a lot of the resources that we already have here at Duke, work that many of us are interested in, and basically saying, can we identify biomarkers of cardiovascular disease in HIV-infected individuals? And so our hypothesis was that we would be able to use a biorepository that's already existing here at Duke, thanks to the efforts of, I'm sure, many people in the room, and Susanna Nagy and Mary McGill are kind of taking the leads of that now. And could we identify novel biomarkers? And those would be metabolic biomarkers, kind of building off the ER stress work with the very focused hypothesis that if these metabolites are reporting on ER stress, and there is potentially a role of ER stress either in primary HIV or HIV medications, that perhaps these ER stress metabolites actually will predict cardiovascular disease. And as a sort of parallel aim to look at echocardiographic biomarkers, we have some really new ways that we can look at echoes. These are cardiac ultrasounds that are early biomarkers of cardiovascular disease and may be better predictors of who's going to develop cardiovascular disease in HIV infection. And so just to kind of quickly go through our specific aims, specific aim one was to find these new biomarkers, and capitalizing on this Duke HIV database, and thanks to the efforts of Lance Okecki, we identified 524 HIV-infected individuals from the Duke HIV database who have been followed for cardiovascular events, and we were able to create sort of a case control, nested case control set of individuals. And what we'll do in these individuals is then do really sort of dense echocardiographic phenotyping, looking at their echoes, looking at these new markers that we have on echoes. So patients whose echoes are really normal, supposedly, if you look at the clinical read, but if you really kind of dig into the echocardiographic biomarkers, that we may find something that is associated with cardiovascular disease. And then to do metabolomic profiling also to identify metabolic biomarkers. Aim two, sorry for all the words, but aim two is really then to say, well, aim one was to look at baseline things. Aim two is really because a lot of these people have serial blood samples and serial echocardiographic studies that we can look at how these things change over time. Wouldn't it be interesting if some of these ER stress biomarkers do change with HIV medications? And this is smaller data set within that 524, really an exploratory aim. But if we found a signal, it would be provocative. As to our study population, I'll kind of skip over this other than to note that we do have a non-HIV infected sort of, I don't know if you want to call it a control group, but that was felt to be an important component of this. We had to pick that from a different biorepository, but we do have a comparison group of non-HIV infected individuals. And I'm actually just going to kind of wrap up with what I hope this will lead to is there's another one of the RFAs through the NHLBI is an R1 RFA, which we do in January. So we're actually proceeding, thanks to, again, through efforts from my colleagues in infectious diseases, to go ahead and do a small pilot study, even sort of waiting for our R21 to find out whether it gets funded, so that we have some preliminary data, actually put in an R01 for the January submission, where we would look at these metabolic biomarkers in a larger set of the HIV database, not just a nested case control, understand how biomarkers change over time, and then also do a sort of in vivo aim, hopefully with Dr. Klotman, where we could do cell-based studies and understand how these metabolites change with primary HIV infection and with medications. So, sorry, I went over as I always do. Thank you. Do you guys have any questions? Dr. Klotman.
Very exciting. It's possible
That's a great question. So we don't have packaging tubes to be able to look at RNA transcripts. The R21 would also sort of allow us to fund like a cytokine panel and be able to look at some of the inflammatory biomarkers. What I can say is that the non-targeted metabolomic platform, so we do targeted metabolomics, which would sort of report on these ER stress biomarkers, but the sort of unbiased metabolome-wide approach doesn't specifically target any of the inflammatory biomarkers, but does provide you a little bit of an agnostic view. And again, hopefully for the R01, I think it would be important to include a lot. And I understand there's a big role of monocyte activation, I think, potentially in HIV, and so we definitely have to address that. Yeah, yeah.
And if so, then you might think that if an HIV-infected individual, some of these metabolites accumulate, that they could actually play a cardiovascular.
Yeah, no, I think it's a really good point, and I think we can do some of that stuff statistically. We have so what I would say from the genetic standpoint is if you believe that there are potentially genetic and epigenetic variation that predisposes, right? So this is, DNA-based variation that predisposes you to proteasome dysfunction, then you would suspect that comes earlier. But I think in the end, you have to do the in vivo studies to really prove that, yeah.
Can you model that in a mouse?
Yeah, you could model it in a mouse, yeah. And you can actually give short-chain dicarboxylacyl carnitines. I mean, you can make them and give them and look at mouse atherosclerosis. Yeah, it's a great idea. Definitely stuff we should move forward with. Oh, really? That would be very interesting. No, I think I think the chicken egg, I do statistics, right? So the chicken egg is really difficult on the statistical side. It always takes you sort of more fundamental people to really establish causality.
Okay. Thank you very much.
Thanks.